In this session, we will learn how to evaluate nucleic acids from the structural perspective. Future sessions will cover how to evaluate nucleic acids from the sequence perspective. We will be using an application provided by DNA Star, a commercial bioinformatics application suite, under their educational license. Nucleic acid analysis can include a variety of common tasks, including searching for motif patterns, analyzing GC content, identifying repeat elements, evaluating potential RNA folding patterns, and seeking transcription factor binding sites. In this session, we'll use an application called GeneQuest, which is particularly suited for nucleic acid structural analysis. We'll open our GenBank sequence that was identified in a previous exercise. And again, we have to change the file formats and pick all formats. And then we can see our file listed. So we'll select that file and click Open. Then GeneQuest will open the file and display it in this pane. There's a couple of things we can do here. First of all, we can see that we can zoom in and out. We can evaluate the GC content by selecting this window and dragging and dropping that. And it shows a graph displaying the GC content. And again, we can zoom in and out. And when we zoom in further, we see the actual DNA sequence. Now, let's select a portion of the sequence and attempt to view it as a potential RNA folding pattern. So you can see I've selected a large part of the sequence and then go to Analysis and pick Fold as RNA. But you can see that the selection I chose was too long. So let's try again, this time with a shorter region. So again, we'll keep the cursor select to the arrow, and now we'll choose a much smaller portion of the nucleic acid sequence. So we'll select that, go here, and pick Fold as RNA. And now we can see a nice prediction of potential RNA folding patterns using this application. So you can trace along the sequence and see where the application predicts structures such as stem and loops and other patterns common in RNA folding analysis. In this session, we learned how to evaluate structural aspects of nucleic acid sequence. Future sessions will cover techniques to analyze the contents of the sequence itself.